1 second.
Okay.
Actually, it was not, recorded. So that's the reason why I had a doubt. Now they opened it. Okay, now they will continue with the class. No problem. Okay. Now, next topic is gene
Gene
transfer
transfer.
methods in prokaryotes. Okay. Now, before we go to the gene transfer methods in prokaryotes, you all know prokaryotes are nothing but examples: bacteria, then fungi, viruses, and all those things. Okay. Now, here we should know about what is actually recombination. We have already talked about what is recombination, what is linkage, and all those things. So, here exchange of the genes between two DNA molecules, okay, to form new combinations, we call it as nothing but a genetic recombination. Now, recombination actually is a very, very important phenomena. Now, because of recombination only, the new combinations of genes will be formed, okay, and it contributes to the population's genetic diversity. So you will get more number of what do you say, strains, or more number of species you will get it because of recombination. Linkage is totally different, so don't get confused. Get confused linkage with the recombination. Linkage is nothing but tendency to remain together during the transmission, but recombination is what? Where actually during the crossover phenomena, exchange of genes will be taking place between the two DNA molecules to form new combinations. Okay, and it is more likely than mutation, so to be beneficial. So it is actually destroys. I mean, less likely destroy a gene's function and may bring together a number of combination of the genes. So that is what is called as genetic recombination or the gene recombination. Okay. Now coming to the gene transfer methods in prokaryotes. So actually, the gene transfer is nothing but the transfer of the genes or the transfer of the chromosome from the parent to the offspring, or from, between the what do you say, two DNA molecules or whatever. So basically speaking, the gene transfer, we have two different methods of gene transfer are there. One is called as a vertical gene transfer, and the other one is called as a horizontal gene transfer. Okay. Now, vertical gene transfer actually will be from parents to the offspring. Vertical gene transfer means from the parents or to the offspring, whereas horizontal gene transfer involves from one microbe to another. Means say bacteria to bacteria or bacteria to virus or whatever it is. So from one microbe to another. And it is between different strains and species of bacteria and the viruses. And it leads to what? Recombination. So these are the basically there are two methods of gene transfer. One is called as a vertical gene transfer, and the other one you call it as a horizontal gene transfer. Vertical gene transfer is between the parents to offspring, and horizontal gene transfer is between the microbes, means from one microbe to another, means between the bacteria. It may be different strains of bacteria or between bacteria and virus or whatever. So horizontal gene transfer actually leads to recombination. Okay, remember that. Okay. So this is then. Now we'll see what is horizontal gene transfer, because horizontal gene transfer actually is very, very important. So here a part of a total DNA from the donor cell will be integrated or incorporated into a recipient cell. Then the remaining amount of DNA, whatever is present from the donor cell, it will get disintegrated, it will get degraded. And the recipient recipient cell with the DNA from the donor, you call it as a recombinant. And so 1% of the population might undergo recombination. So that is actually the concept of horizontal gene transfer. Okay. Now in horizontal gene transfer, what is there? It is nothing but the transfer of the gene between one microbe to another, or between bacteria and virus, or between two states, or between two different species. Okay. So here horizontal gene transfer is the part of total DNA, okay, from the donor cell, which is incorporated into the recipient cell. So remaining amount of, so when once it is incorporated into the cell, remaining amount of DNA will be there, no, from the donor cell, it will get degraded, and the recipient cell with the DNA, okay, from the donor, you call it as a recombinant. And from this phenomena, only 1% of the population will undergo the recombination. Okay. Now, horizontal gene transfer mechanisms actually are of three types. One is called as transformation, transduction, and conjugation. So now we are talking with reference to only prokaryotes, because in your syllabus you have got gene transfer mechanisms or the gene transfer methods in prokaryotes. So the recombination in prokaryotes occurs through three methods or through three mechanisms. One is called as the transformation, then you have the transduction, and the third one is nothing but the conjunction. Okay, now we will see one by one. Now what is transformation? Okay, now transformation. Now, actually, transformation actually was one of the earliest mechanisms. So among the three, it was the first mechanism or the earliest mechanism that was being described, and it was first discovered by Frederick Griffith in 1928 in Streptococcus pneumoniae. Okay, so it was first discovered. He discovered this particular experiment or this, okay, in the Streptococcus pneumoniae, and he showed that the DNA is the genetic material and it can be transferred from between a donor as well as a recipient cell. So what does transformation says? It is nothing but the transfer of the naked DNA from the donor to the recipient cell. That is what is called as the what transformation. So the transformation is a phenomena or a gene transfer mechanism which is seen in the prokaryotes, and it is the first mechanism actually which was being discovered, and it was discovered by Frederick Griffith in 1928 in a bacterium called Streptococcus pneumoniae. And what does it say? Transformation definition is nothing but transfer of the naked DNA from the donor to the recipient cell. So he did this experiment in Streptococcus and he showed that the DNA actually is a genetic material and it can be transferred between a donor as well as a recipient cell. Now, later on, Griffith's experiment, what he told is, Griffith, a virulent Streptococcus pneumoniae became virulent when exposed to the heat kill virulent cell. So that is what his finding was. Now we will see how actually he has performed the experiment. So this is the transformation experiment actually that he performed by Griffith. Okay. So what is transformation? It is nothing but the transfer of the naked DNA from a donor cell to a recipient cell. So here, see here, okay, the bacteria with the prokaryote cell, living virulent, then here, heat kill non-virulent, then here, living non-virulent, heat kill non-virulent. So like this, okay. Now we'll see the mechanism now over here. Okay. Now bacterial transformation without the mice. So here they are not involved mice in the experiment. The figure they have shown, but here they are explaining the bacterial transformation without the involvement of mice. Now we will see. So the broth containing the non-encapsulated living bacteria and dead encapsulated bacteria are incubated. Okay. So what do you mean by this? You know what is a broth? Broth is nothing but a media. It is a liquid media. So what does it contain? It contains what? The living bacteria. And they are non-encapsulated. Non-encapsulated means removal of capsule. They don't have, they are naked. So they are non-encapsulated. So when the bacterial culture which contains or the non-encapsulated means it has, what do you say? It has got the cover. Okay, living bacteria and dead encapsulated. Okay, they are dead and encapsulated bacteria are incubated. So what happens? After incubation, encapsulated living virulent bacteria are found. So non-encapsulated bacteria will receive the genes from the dead encapsulated and forms a capsule. So the material responsible for the transmission of this character was not known actually, basically. Okay. So what did they do? How they perform this experiment? Transformation is what? Nothing but transfer of the naked DNA from the donor into the recipient cell. So without involvement of mice, how they perform this experiment? This is actually the experiment which has been performed by the Griffith. So he took a broth containing what? Non-encapsulated living bacteria. Okay. Okay, and they have incubated, means together they try to incubate it. After incubation, they could observe the encapsulated living virulent bacteria. Then later on, this encapsulated bacteria will be there, no? They receive from the dead encapsulated or formed a capsule. Next, now experiment. So here, this is DNA is the genetic material. That is how they prove. This is how they have taken. Now, another experiment in 1944. Oxford, Avery, Pauline, and all those things. They try to prove that DNA is the genetic material. So actually, in this experiment, they try to prove that genetic material is nothing. DNA is the genetic material. So what they have done is they have taken a mixture of DNA and RNA, treated with an RNase enzyme, and when they treated with an RNase enzyme, RNA actually got degraded, only the DNA remains. Okay. And that DNA actually they have added to the bacteria. Okay. When they have added to the bacteria, they have plated on the growth medium. Say here C. That is, then they could produce these transformants. So when once these transformants were produced, then next is when they treated with the DNA, only RNA remains. So what they did here, RNA DNA is there, and here a RNA will be there. So that RNA
Yes.
they have added, okay, to the bacterial culture, and when once they added to the bacterial
Share
culture,
the family.
the RNA and then they have plated. So there were no transformants. So that means by performing this experiment, they try to prove that DNA is the genetic material, but not RNA. Are you understanding?
अरे ना
Okay.
क्या पागल है जी, इनको कुछ भी काम करे देता नहीं,
That
पूरे
is
क्लास
that
में
is what
शिफ्ट
they
कर
have
दिए.
tried to prove. So this was actually proven by Oswald, Avery, and Collins. So they tried to prove that DNA is the genetic material.
झगड़ा लगा रहे थे.
Now we will see how actually. The next is what? Competence. What do you mean by competence? Now, competence is nothing but the ability of a recipient bacterium, okay, to take up the DNA from the environment. Competent cells are what? The cells which can be transformed. And E. coli cannot undergo transformation naturally. E. coli cannot go. So what it has to do? It has to be made competent by artificial transformation.
अलग जगह लगाते थे. मेरे को
Okay?
बोलना था,
Right?
तुम चुप करती थी,
So you have
बताओ.
to induce artificial transformation because it does not undergo the transformation by natural phenomena. So it has to be done by artificial transformation. So this is all regarding the transformation experiment. Remember, students, okay? So competent cells and competence. So competence is what? Nothing but the ability of a recipient bacterium, okay, to take up the DNA from the environment. And competent cells are what? Which can be transformed. See, when once you take any cell, you have to actually get it transformed. Means you have to form a new one. Okay. Then E. coli cannot undergo the transformation on its own. Means bacteria cannot do. So it has to be induced by artificial means. Okay. So that is what they are saying. Now, mechanism of transformation. Now we will see how they have done this. So after the death, cell lysis will lead to the release of DNA from the bacteria. So other bacteria will take up the DNA and then they will integrate into their chromosomes by recombination. The recipient cell with this combination of the genes will now become a hybrid or a recombinant. Okay. So this is transformation in nature. Actually, by natural phenomena this is, but by artificial phenomena only E. coli. So by natural phenomena, the mechanism of transformation can be seen in bacillus, in Haemophilus, Streptococcus, Staphylococcus, as well as the Neisseria. Are you understanding? What is transformation? Transformation is nothing but what? It is the transfer of the naked DNA, okay, from the donor into the recipient cell. So there are two ways they are saying. One is called as, one experiment we have seen how DNA actually is works best as a genetic material when compared to that of RNA. Then they try to explain over here that DNA is a genetic material. Next, they try to explain over here that the transformation cannot be, I mean, there are two methods of transformation we can say. One is by natural method and the other by artificial method. Artificial transformation can be seen or can be done only in E. coli because it cannot transform. E. coli cannot be transformed naturally. So that is why their transformation has to be done only by artificial means. Whereas in the natural phenomena, you can observe it in Bacillus, Haemophilus, Streptococcus, Staphylococcus, and Neisseria. So here it is very simple. So what happens is after death, the lysis of the cell will take place. So when once the cell lysis, the DNA will be released from the bacterium. The other bacteria will be there, no? They will take up the DNA and then they will integrate into the chromosome by recombination phenomena. So when once the recipient cell take up this new combination of the genes, it will undergo the transformation and it will become a hybrid or a totally a new recombinant. Okay. So this is what they're saying. Okay. Then now we will see step by step how actually it will take. So now the DNA binding receptor on a competent bacterium will bind the double-stranded DNA. Then as the DNA enters the cell, one strand is degraded and the other strand is coated with single-strand DNA binding protein. Are you understanding? Students, are you all listening? Yes? Hello? Hello?
Yes, ma'am. Understand, ma'am.
Yeah. So here now I'm talking the mechanism, how the transformation will take place. You know what is transformation now? Okay. Now I have talked about artificial transformation and I have talked about the what natural transformation. Natural transformation is seen in different species, but artificial transformation can be seen only in E. coli. Now we will see what is the mechanism of transformation, how actually the transformation phenomena take place in the bacteria. So here what they're saying is the DNA binding receptor on a competent bacterium will bind the double stranded because when once the cell lysis, we are saying that the DNA will be released. So that DNA actually will be taken up by other bacteria. So here the DNA binding receptor on a competent bacterium. Competent bacterium here is what? The one which takes the bacteria, okay, or the recipient bacterium you can say. So it will bind the double-stranded DNA. So as the DNA enters the cell, what happens? DNA is double-stranded, you all know. One strand will get degraded and the other strand actually will be coated with a single DNA, strand DNA binding protein. Then in the second step, what happens? The single strand of the donor DNA then is integrated into the chromosome of the recipient cell, producing a recombinant DNA. Okay, so now one strand only will remain. So that single strand of donor DNA, one will be degraded, no, and another only will be there. So this single strand of donor DNA will get integrated into the chromosome of the recipient cell, and it will produce a recombinant DNA. So this is how the mechanism. Okay, the transformation with DNA fragments. The DNA fragments will be there. It will get integrated into the bacterial chromosome. Then uptake of DNA will take place. See here, or degradation will be there, unsuccessful. But here integration by non-reciprocal recombination like this. Okay, and transformation with a plasmid also we can do. We can also insert it that DNA with the help of a DNA plasmid. So that bacterial chromosome with the help of the plasmid, you can insert it into another chromosome, then uptake of the plasmid, and you get a stable transformation. Okay. Now this is about the transformation. Now coming to conjugation. Now what do you mean by conjugation? Now here, the conjugation is what? The transfer of the genes between the cells that are in physical contact with one another. Okay, conjugation means just by physical contact. Okay, when the genes are being transferred, then you say it is a conjugation. Okay, transformation is totally different. Conjugation is nothing but the transformation of or the transfer of the genetic material from cell to cell by direct contact. It is by direct contact. You call it as nothing but the conjugation. Now, a conjugation, actually, the best example you can give E. coli. Okay, so here transfer of the genes between the cells that are in physical contact with one another, you call it as a conjugation. So it was first demonstrated in the bacterium by Lederberg and Totem in the year 1946. And they found that the genetic traits could be transferred among two different strains of E. coli if they are in a physical contact. And this experiment actually they tried to perform it in E. coli, and this was given actually basically by Lederberg and the Totem. Okay. Now, later on, William Hayes, Francis Jacob, and Ellie Woolman. Okay, they try to actually they could find out two factors are responsible. See, when they are in close contact, means there should be some factors responsible for the for the mating or for coming close together or for attachment purpose. So the William Hayes and Francis Jacob and Woolman, they actually try to give the two factors that are responsible for this conjugation phenomena are nothing but the F plus and the F minus factors. So based on this, actually they said that the conjugating bacteria actually are of two mating types. One is called as the F plus cells and the other one you call it as the F minus cells. These F plus and F minus cells, they are called as the factors, or that is the fertility factor you can call them, or the F minus factor is called as a sex factor. So F plus and F factors are very, very essential for the mating phenomena. So here the male types which donate their DNA, okay, they are called as what? The F plus cells. Whereas the female types which are recipient of the DNA donated by F plus, they are called as nothing but the F minus cells. So these F plus and F minus cells, they are called as the fertility factor or F minus factor or the sex factor. So remember, for the conjugation phenomena, you require two factors. What are those? F plus and F minus. Now, these F plus and F minus factors which are responsible for the conjugation phenomena actually have been given or proposed or have been found out by Hayes, Jacob, and Woolman. Okay, so based on this, actually F plus cells are what? Nothing but the male types. Okay, which donate their DNA. Whereas the female types donate recipients. Females are the recipients, whereas the males are the donors. So we can say males are the F plus cells and females are the F minus cells. So in that case, F plus cells you call fertility factor and F minus you call it as a sex factor. Okay. Now, how this process of conjugation phenomena is taking place, we'll see. Okay, they are also called as sex pili. You might be okay. So the F pili of the F plus donor cell will make contact with the F minus recipient cell and will pull the cell together. So just now I've told that there should be some mechanism, some the factors should be there which should allow them or make them attract to come close together. Then only the mating will take place. So here the F pili, which is also called as the sex pili of the F plus donor. F plus donor here is nothing but the male cell. Okay. F plus donor cell will make contact with the F minus recipient cell, which is the female, and it will pull the cell together. So what happens when once they come together, rolling circle replication transfer one strand of the F factor into the recipient cell. So what happens? When they come close together, the replication in the form of circle, that is what is called as the rolling circle replication, will take place. So the transfer they transfer one strand of the F factor into the recipient cell. Okay, so when once the transfer of the F factor is completed, it will yield 2 F plus factor bacteria. It will yield how much? 2 F plus factor bacteria. Okay, so this is how the F process of conjugation will take place. See here, this is the F plasmid. Okay, so this is the conjugation file, as I told you, that is what is called as the sex pili. This is another chromosome. So donor cell attaches to a recipient cell with its pilus, and the pilus draws the cells together. So this is F plus cell, this is F minus cell. So the cells actually will get, with the help of sex pili, they come close together and they contact one another. Then next what happens? One strand of the plasmid DNA will transfer to the recipient. Then the recipient, what it will do? It will synthesize a complementary strand to become a F plus cell, and the donor will synthesize a complementary strand, restoring its complete plasmid. So that is how the process of conjugation will take place. Okay. So the next what happens is in donor F plus cells. In donor F plus cells, what happens? The F factor may integrate into the host chromosome, becoming HFR. That is what is called high frequency of recombination. Thus, the F plus cells will become the HFR cells, and later the conjugation between HFR and the F minus cells will result, and it will form, or it will result in the replication of the chromosome with the F factor. Then later on, a single parental strand is being transferred from HFR cell to the F minus cell. So this is all the various steps actually that are involved in the conjugation process. So first, step by step, we have seen what is happening. So actually, for the mating phenomena, what happens? You require two factors for the conjugation. One is called as F plus factor, and the other one is called as F minus factor. F plus factor actually is a male one, and F minus factor actually is a female one. So when they have to come close together, means the F factor, means the sex pili, which is present on the the donor cell, that is the F pilus of the male. Okay, it will try to attract the female. So as a result, what happens? The sex pilus come close together. They try to attach. So when once they get attached, then what happens? The one strand of DNA. The donor here is the male. So from the donor, from the male, the what happens? The DNA will be integrated, or it will be transferred to the female. So like this, actually. So now here the donor is the F plus cell, which is nothing but a male, whereas the recipient is the female, that is F minus. So in the donor F plus cells, what happens? The F factor will integrate into the host chromosome, becoming HFR. Okay. Then HFR is what? Nothing but the high frequency recombination. So these F plus cells, what happens? They will become HFR cells. And when once they become HFR cells, the conjugation between HFR and F minus will result in the replication of the chromosome. So as a result of this, a single parental strand will be transferred from HFR cell to the F minus cell. Okay. So this is how the process of conjugation, just now I've already told you. Okay. So you should clearly understand what is F plus, what is F minus, what do you mean by HFR, and all those things. So remember two factors, F plus and F minus. Okay. With the help of sex pili, they come close together. Donor here is F plus, which is a male, and F minus is a female. Okay, which is a recipient, and with the help of this plasmid, actually how they get integrated, and how actually the sex pili will help them. And once it enters, then what happens? This F plus will become HFR. So when once this F plus will become HFR, then what happens is that there will be a recombination phenomena will be taking place between the HFR as well as the F plus cell. So when once it takes place, then that HFR will be integrated, or it will be transferred into the F minus cell, that is into the female cell. So that is how actually it will take place. Okay. This is the process of conjugation. Okay. Then what happens? Complete transfer of the chromosome does not take place here, they are saying. Only a small piece of the F factor will lead the chromosomal genes into the F cell. See, complete transfer of the chromosome. See, the chromosomes actually will not go whole, but only in parts it will go. Only a fraction, you can say, or only a small piece of the F factor will lead to the chromosomal genes into the F minus cells. The complete transfer of the chromosome will not take place. So the small strand containing these chromosomal genes, it will recombine with the DNA of the F cells. So that F cells will receive only a part of the chromosomal genes and hence do not be converted into the F plus cells. They do not be converted into the F plus. I understand. So this is how actually you should remember. So the complete transfer of the chromosome from the F plus, that is the donor, will not be taking place. Only a part of it will be taking place. Okay. And this small strand which contains this one, it will recombine with the DNA of the F minus cell. And thus the F minus cells will receive only part of it and hence they do not get converted into the F plus cell. Okay. So this is about the, what is that? Conjugation. Now coming to the transduction. Now transduction actually is the last mechanism which we see in the prokaryotic organisms. So here transduction actually occurs when a phage, that is a virus, carries the bacterial genes from one host cell to another. Here now the virus will play an important role over here. So there we have seen only between two bacterial strains. Okay, the conjugation as well as the transformation we have seen there. But here in case of transduction, you require a phage particle, that is nothing but a virus. So transduction always occurs between a virus. So when a phage particle, that is when a phage virus carries the bacterial genes from one host cell to another cell, then we can observe the transduction phenomena. So that is what is called as nothing but the transduction. Okay. Now this transduction actually was described by Norton, Zinder, and Joshua Lederberg. Okay. It was discovered by Norton, Zinder, and Joshua Lederberg. So phage is nothing but a bacteriophage, where we call it as a T4 lambda phage bacteria. So they have explained this particular phenomena in this lambda phage virus, and it occurs, they are saying that only when this phage particle carries a bacterial gene from one host cell to another cell. Now we will see the mechanism. Okay. Now when you talk about this transduction, okay, now here you have two types of cycles you will come across in case of transduction phenomena. So transduction actually, it is a third mechanism that we see in bacteria, and two different types of transduction actually have been observed. Now before we go to that, actually we should know what is the lytic cycle and what is the lysogenic cycle that we observe. Okay. Now coming to this lytic and lysogenic cycle, bacteriophage attaches to the donor bacteria. Then it will try to inject their nucleic acid into the bacterium. Then DNA will replicate rapidly and also direct the synthesis of new phage protein. Then a new DNA will be formed. It will combine with the new protein to make a whole phage particle. Then they are released by the destruction of the cell wall and the lysis of the cell. So that is what is called as the lytic cycle. Okay. Lytic as well as the lysogenic. So lytic and lysogenic cycle is very, very important to understand the concept of the transduction mechanism. So here what they are saying is bacteriophage will attach to the donor bacteria. Then they will try to inject that DNA into the bacterium. And once they inject the DNA into the bacterium, DNA of the bacterium will replicate rapidly and also direct the synthesis of other phage proteins. Then what happens? The new DNA that is there will combine with the new protein and it will form the whole phage particle. So once the whole phage particle is being formed, phage particle is nothing but here is a virus particle. So then they are released by the destruction of the cell wall and the lysis of the cell. That is how the lytic and the lysogenic phenomena will be taking place. Okay. Now coming to the process of the transduction. Now, as I have already told you, transduction actually is again a third mechanism which you observe in case of the bacteria. So these phages may be composed of DNA of the host in this case, because what is the transduction phenomena? Transduction phenomena actually here nothing. The bacterial genes are incorporated into a phage. Okay, Here the bacterial genes. So when means that when the phage bacteria actually carries the bacterial genes, okay, from a host, then you call it as nothing but a transduction phenomenon. Already have explained it to you. Okay. So in this case, what is happening? These phages are composed of what? DNA of the host. Then the phage will attack the another host and it will infect it. So here the phage particle here is nothing but the viral DNA. Okay, they consist of what? The DNA. They consist of the DNA of the host cell. The host may be say bacteria, and this phage will when it attacks the another host and it will try to infect it. So when it infects it, what happens? The recipient DNA will integrate into this DNA. The recipient DNA will integrate into this DNA and it will result in the transfer of DNA. It will result in the transfer of DNA. So now the recipient cell is now called as the transduced cell. Means the recipient cell, what it is trying to do? It is trying to actually totally get transformed into a new cell. Transfer of. So that is what is called as the transduced cell or transduction. So what is the transduction phenomena? Transduction phenomena is nothing but here when a phage carries a bacterial genes, okay, carries the bacterial genes of a host, and then when they are incorporated into the another host, then that process you call it as transduction. So this actually involves a phage particle or a virus. So these phages are composed of DNA of the host, and these phages, what do they do is when they attack another host and infect it, the recipient DNA will get gets integrated with this DNA, and it will result in the transfer of DNA. So now the recipient cell which receives this DNA, you call it as a transduced cell. Okay. Now, basically, there are two methods of transduction. They are called as generalized or non-specialized transduction, and the other one is restricted or specialized transduction. There are two types. Now, generalized transduction and restricted transduction. Now, generalized transduction, in this case, all fragments of the bacterial DNA have a chance to enter a transducing phase. So, in the generalized form, transfer of, okay, they can transfer any segment of the bacterial genome to another bacteria. They can transform any segment of the DNA, or you can say all fragments of the bacterial DNA actually have got the chance to enter into the transducing phase or to enter into a bacterial phase. Whereas in case of the specialized transduction, okay, then what happens here? Certain phages only can transfer only a few restricted genes of the bacterial chromosome. So that is why it is not called as a generalized one. Here, only few phages, certain phages can transfer only a few restricted genes, okay, of the bacterial chromosome. So only those bacterial genes adjacent to the prophage to the bacterial chromosome. So it mediates the exchange of only limited members of specific genes. And this is done by the bacteriophage lambda. And this is done only by the bacteria. So actually what is specialized transduction is, it is only restricted segments of the bacterial genomes are being transferred. Whereas in case of specialized or generalized one, it can transfer any segment of the bacterial genome. There is no restriction at all. But in case of specialized one, there is a restriction. So only restricted segments of the bacterial genome can be transferred. And it is being done only by the bacteriophage one. Okay, this is the specialized transduction. So lambda phage can transfer. Okay, only incorporate into a specific site. So all gene is on one side. So okay, so this is the specialized transduction. Okay, so this is all about the what do you say, the different mechanisms of gene transfer. Okay, so students, is it clear? Hello, 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 hello. Are you able to understand? Hello, hello. Are you able to understand, students? Hello. Okay, so this is what actually we have discussed today, the gene transfer methods, the transfer methods in the bacteria. Three different types I have already mentioned to you. One is called as the transformation, conjugation, and the third one is the transduction. So these are actually the three different types of gene transfer mechanisms, and it comes under the horizontal gene transfer methods. In the horizontal gene transfer methods, the recombination will take place. Whereas in case of the vertical gene transfer, there is no recombination, and it is nothing but the transfer of genes from the parent to the offspring. But here in this case, with reference to the prokaryotes, you will observe only the horizontal gene transfer mechanism, where it is of three types. One is called as the transformation, transduction, as well as the conjugation. Okay, so this is about the different gene transfer methods or the gene transfer mechanisms in the prokaryotic organism. That is about the bacteria. Is it okay, students? Hello, are you listening? Yes. Hello.
Yes, ma'am.
Hmm. So, are you able to understand?
Yes, ma'am. Yes, ma'am.
Okay. So, this is what we have done today. Okay. So, next class we will see the other chapters. Okay. Now it's already time. Now we will leave the meeting. Okay.
Mhm.